Okay, so welcome back to our 501 lecture series, Genetic Information Transfer. In today's lecture, we're going to be learning about large-scale genomic rearrangement. So these are going to be things like double-stranded breaks, so entire chunks of chromosome being broken, possibly even lost, and recombination, the mechanism by which many of these double-stranded breaks are repaired. And we'll learn about other types of recombination that occur inside the cell with things like transposons and things like that. So whereas the previous lectures we thought very narrowly about DNA damage and DNA repair and DNA modification, here we're going to be thinking about very large chunks that can be used as templates or be moved and inserted to other places in the genome. And so just to remind you about what happened last time we talked about chemical damage to DNA bases and their repair. So we're thinking about very specific types of damages like alkylation, deamination, depurination, or UV dimers that occur at very specific sites in the genome. And we talked about the different repair mechanisms that exist to fix those damaged sites. And so this usually involves excision or removal of either the base or the stretch of DNA where the problem has occurred, and then resynthesis of that stretch. But the types of damage that can occur, so if you went into outer space or some gamma rays blasted you in the face, another type of damage that can occur that's difficult to repair by any of the mechanisms we described so far are double-stranded breaks. So imagine something happens where an entire chromosome gets split right down the middle, so broken, say, into two pieces. As the cell, it's going to need to repair those two broken pieces. It's going to have to somehow put them back together. Otherwise, you're going to have the wrong number of chromosomes. You're going to have problems with segregating your chromosomes and replicating them properly during division, and that can lead to all sorts of catastrophes. So we definitely want to fix this. And so in this lecture, we're going to learn about how chromosomes can be broken and repaired by recombination, as well as how recombination more generally can be used to insert or remove large pieces of DNA at genomic sites. So that's what we're going to be talking about today. As usual, I'm putting the lecture objectives here, so you want to be, as you're listening to these lectures, paying close attention to these types of topics. In particular, we're going to be talking about homology-directed recombination. We're going to spend a lot of time on that. That's the main way in which cells repair these double-strand breaks. And then after we've learned about that, we're going to talk a little bit more about sequence-specific, or what are called site-directed recombination events that are used to perform recombination at very specific locations in the genome. And these are often associated with viruses or in selfish genetic elements that can sort of hop around and move themselves in and out of the genome. And those activities will make more sense once we've talked about homology-directed recombination. So let's just dive in and let's talk about the problem that the cell needs to be able to solve. So let's say your chromosome breaks. And when I say your chromosome breaks, what I mean is we have this lovely chromosome here, and there's some stretch of DNA, 5' to 3', 3' to 5', and something happens, this lightning bolt here, which could be gamma rays, could be X-rays, any of these things that can induce what's called a double-stranded break. And that is a clean break of both strands, a cut that goes all the way through the DNA to generate these two fragments like you see here. And so if you're the cell and this has happened to you, what do you do to repair this damage? And so the first thought that probably comes to mind is, well, why don't I just ligate the two pieces back together? You learned about DNA ligase activity. As long as we can connect this 3' end to this 5' end and this 5' end to this 3' end, then we should just be able to stitch these two pieces back together and done. We're problem solved, right? And that sort of would make sense if this break, we were sure that this break happened and we immediately caught it and the two pieces were right next to one another. But more generally inside the cell, how can you be sure that you're fusing the right ends together? How do you know you're connecting the right pieces to one another? Because what if there are other free ends inside the cell? Or what if you had two double-strand breaks and a whole chunk of the chromosome went missing, and now you stitch two pieces together and you delete everything? You might accidentally fuse two regions together that shouldn't be connected. And so hypothetically here, this is a scenario where we want to connect the pink pieces, but maybe the cell would accidentally connect the purple piece or the gold brown color pieces together here, and now we've got the wrong things, right? So is there some way that the cell can repair these types of double strand breaks with more confidence so that it can be very sure that it's repairing these breaks in the right way and not accidentally making these purple pink mashup strands like you see here? And so the cell has a clever solution to this, which is to remember, first of all, that your chromosomes come in homologous pairs. And that means for every chromosome that you got from your mom, you got one from your dad as well. And in principle, they have more or less the same information on them. Obviously, your mom's genes are different from your dad's gene, but more or less, your mom has some version of every gene, your dad has some version of every gene. And so if a break happens in one chromosome, you could imagine that you can use the other copy as sort of your guide for how to reassemble the broken pieces. And it turns out this is exactly what the cell is going to do. And the idea here is if you've got some break, so you had some chromosome that broke, and there's some red piece on one side of the break and some of this blue DNA on the other side of the break, well, you could look for a stretch of DNA on your homologous chromosome that sufficiently matches on both sides of your break. And then you should just say, well, whatever segment is in between those two, that's what I should be repairing my break with. And this might mean actually that you use huge amounts of your homologous chromosome to make the repair. And you're making that inference based solely on the flanking sequences surrounding that homology. So I hope that's clear that if you have a break and you've got these red strands and these blue strands on either side of your break, the cell's going to look for a stretch of DNA that has blue and red on those sides, and then assume that whatever is in between is what it should place in there. Okay, so that's the idea, and now we have to figure out how the cell is actually going to implement this strategy. And so if we think about what needs to happen here, if we have these breaks in one chromosome and we want to repair with another, if we want to make use of the idea that the red strands on one chromosome match the corresponding strands on the other, then we need an opportunity to do Watson-Crick base pairing, Watson-Crick flanker base pairing. And so in order to search for homologous regions, we're going to have to chew back one of the strands to expose a single-stranded overhang. And that's because we need a single-stranded piece of DNA to be able to go exploring for a partner sequence that matches it. And in the cell, it is a 5' to 3' exonuclease that generates these 3' overhangs. Now, in principle, you can imagine either a 5' to 3' exonuclease generating 3' overhangs, or a 3' to 5' exonuclease generating 5' overhangs. Both of those would be ways of generating single-stranded overhangs that you could use to search for homology. But if the reason that a 3' overhang is particularly useful for repair is because once the base pairing between the other chromosome is found, that 3' overhang, you've got a 3' OH at the end, right? And that means that the DNA polymerase can start extending off of it to match, or rather to copy the matching target and basically fill in the information. And so that's why it's really critical for this particular repair pathway to generate these free 3' overhangs. So once you have a free 3' overhang, and it is able, if it is able to make base pairing interactions with a target chromosome, it will start to displace the base pairing strand of that other chromosome. So basically, this 3' overhang slips in and starts making base pairing interactions with the homologous region of the target chromosome, and that's going to displace one of the strands of the target here. And this is a process that's called strand invasion, when the 3' overhang sort of inserts itself into the DNA of the other one. And as I mentioned before, once you've inserted here, you have a 3' end which polymerase can extend off of, and it uses the homology target as the template. So basically, as it proceeds here, it's going to make a copy of all the sequence that is adjacent here to the homology flanks. Okay, and I should also point out that this 4-way structure that you see here forming is called a holiday junction. So this is where the crossover between these two strands, pieces of DNA is taking, or these two fragments of double-stranded DNA is taking place. And these holiday junction structures, I'm showing you here a detailed structure of one so that you can take a look at it. And the key thing here is I want you to see that basically in these structures that you have here, you've got this four-way DNA junction, and there is B-form character to all of the DNA, the double-stranded DNA that you're forming here, except for this small little stretch in between as the strands swap partners. So you've got this blue strand that's normally paired with green, and then it's going to switch to the yellow strand here. And you've got this red strand that's normally base pairing with the yellow that's going to switch to the green. And it's double-stranded everywhere you go, and you just have this narrow region here where the strands are being displaced. And this structure of this holiday junction actually allows for the two strands to be moved in a coordinated fashion with one another, passing one strand from each double-stranded DNA off to the other. And it turns out that helicases actually can operate on this structure to cleanly translocate the position of this crossover structure in a process that's called branch migration. Okay, and as this process of polymerization and strand invasion continues, eventually the displaced strand that you've produced up here can start to base pair with the other 3' overhang of your break. So now remember, we have another 3' overhang on this blue strand here, and as this displaced strand gets more and more displaced, eventually it will achieve homology with the 3' overhang of the blue strand, and now it can get extended because this is again a 3' overhang, and so now we start synthesizing in this way. And so between the two strands, we will have successfully rebuilt the yellow sequence that's in between the red and blue ends of the fragment and in the correct order. And so in this way, we used one chromosome's pink, yellow, blue to reassemble the yellow sequence that lived between the red and the blue in the broken strand. Now, if the DNA nicks are sealed, so remember, at this point we still have these nicked pieces here, and so using the activities you've learned about in the previous lectures, these DNA nicks can be sealed, and once this happens, we are left with two crossovers. So we now have two holiday junctions that are sort of holding our two chromosomes together here. And obviously we don't want to stop here, because this would be a position in which our two chromosomes are joined together by these double holiday junctions. And so in order to get our ordinary chromosomes back, the cell has to resolve these junctions. And so what does that mean? Well, if we were to look at a holiday junction like this and say, what do we need to do? Well, we're going to need to cut it and connect some of these pieces back together in order to separate this structure. Well, there's going to be two natural ways that you can do this, right? You could cut across the holiday junction horizontally, like you see here, and then you could connect the red and blue strands together like this. So that would be one way in which you could resolve the structure. Or you could cut vertically through the junction, and then you could seal the green and yellow strands together, or yes, the green and yellow strands together on both sides like you see here. So you can make a vertical cut or horizontal cut. And you should note that the choice of how you cut and seal this holiday junction is actually going to affect kind of the origins of the different parts of the DNA in each chromosome. And so kind of to see where that is, if you apply these holiday junction resolution possibilities to the double chromosome or double crossover scenario, you can see that different possibilities can happen following repair. So for example, if we cut across here and just seal this way, then what we get is basically a clean exchange, a clean exchange of information only in the region at which we wanted to copy and repair the DNA, right? So the black chromosome remains black all the way on its flanks. It's only in this region between the blue and red where information was exchanged. But it's also possible to resolve those junctions in such a way that the entire chromosome arms of these two fragments have actually switched places. So you can see now actually that you've got a blue stretch up here, and you now have basically transferred part of one chromosome arm to the other. And this is kind of an interesting point because we've been talking about homologous recombination in the context of repairing breaks, but recombination isn't something that's just used to repair double-stranded breaks. It also functions to generate variety in the distribution of paternal and maternal genes on your chromosomes. So remember, we were talking about how you got one set of chromosomes from your mom, one set of chromosomes from your dad. They each have different alleles of different genes on them. If you aren't allowed to exchange alleles between the two chromosomes, then when you produce gametes to make offspring, all your chromosomes are either going to look exactly like your dad's or exactly like your mom's. And what you'd actually like to do is to kind of shuffle up some of those alleles. And so recombination provides a way to do this by generating variety in the distributions. And it comes from the fact that recombination ends up being a very useful way of making sure that you can pair maternal and paternal chromosomes together, because they have to have homology with one another in order to form these holiday junctions and these recombination structures. So recombination becomes a natural way to make sure mom and dad chromosomes pair. And then when you resolve those holiday junctions, depending on how you do them, you can get exchange between the different chromosomes, and you can get local exchange, or you can get entire arms of chromosomes swapping places like you see here. And so this homologous recombination provides a way of getting crossing over between chromosomes that take place when you make your gametes during meiosis. So to sum up what we have talked about with respect to homologous recombination, I know there's a lot on this slide, but it's a little bit tricky, so I just want to make sure we go through everything very clearly. Double-stranded breaks that occur in the genome can be repaired through this homologous recombination mechanism, and homologous recombination is something that can occur between any highly similar homologous sequences. So it's not a specific sequence that's being looked for. Any sequence that has complementarity to another can provide the substrate for this recombination reaction to occur. And the reason for that is that homology is tested between sequences by generating these single-stranded 3' overhangs, which can then sample and look for things that look exactly complementary to the sequence that has been produced. And once suitable homology is found, then you basically can use DNA polymerase to extend those 3' overhangs using whatever target you found as the template. Now, during this process, 4-way DNA structures called holiday junctions form, and it's really clear to get a picture of those it's important to get a picture of those holiday junctions in your mind, because they're at the core of this strand exchange process. And then depending on how you resolve them, different gene products can be produced from the recombination process. And this is one of the reasons why homologous recombination also plays a role in generating chromosome diversity during gamete production by allowing exchange of portions of chromosomes between, say, your paternal and your maternal chromosomes. So it's both a repair system, but also a way of exchanging whole chunks of chromosome to generate allelic variety. Now, as we said, homologous recombination can be use any arbitrary homologous sequence to direct recombination, and that's very powerful from the repair point of view and the allelic variety point of view. However, what we're going to talk about in the next series of lecture there are other types of recombination systems in which very specific sequences are what are used to direct the recombination. And so this is something that certain genetic elements or viruses use to be able to insert themselves and excise themselves very cleanly from specific locations of the genome. And in the next video, we will learn a little bit more about some of these other recombination systems and how they work. So stay tuned for that.